Okay, so this lecture 18 is over C. elegans. Its full name is Norhabdidis elegans, but it's just commonly referred to as C. elegans. And this lecture covers techniques and genetics of C. elegans, as well as anatomy and development of C. elegans. Some of the highlights of C. elegans in research are that they're small, free-living nematodes and that they are the first animal genome that we were able to sequence. They have a 36% protein match to humans, which makes them good model organisms. They're technically referred to as the best known animal on Earth. Their location and fate of every cell in the adults is known, and development occurs exactly the same pattern for each individual, which is different from other organisms that we talked about because a lot of times we know what groups of cells are doing and what tissues they give rise to, but we don't know the exact fate of individual cells. There's also a defined number of cells in C. elegans adults. They all grow to the same cell number, which is 959 cells, also different from other organisms. We've been studying C. elegans since the 1960s, and they're great for labs because they're crowd preservable, they're small, they have a 3-day generation time, they live around 20 days in the lab. It takes about 10 hours for development from fertilization to hatching, and most notably, they're hermaphroditic and can self-fertilize. So just take a moment and think about why this might be useful for genetic screens. Okay, so since the elegans are hermaphroditic, they can self-fertilize, which means that for genetic studies, we can control outcrossing, and also that means that they can be maintained individually in lines, and we can track all of the genetic lineages really easily. And also, recessive mutations will automatically segregate as homozygotes after just 2 generations, so it's really easy to do mutant screens. And there's no need to set up controlled crosses because they just self-fertilize. We just have to separate the offspring to examine individual lines. Okay, so now we're going to talk a little bit about adult anatomy. Spoiler alert, they're an elongated worm shape that's about 1 millimeters in length, which is just about the size of the tip of your pencil. So they're very small. The adults have 959 somatic cells and about 2,000 germ cells, so they're largely devoted towards reproduction. Another interesting fact about C. elegans is that they grow by cell enlargement rather than by cell division, as most organisms do. So once they reach adult stage, there's no more cell division, and their cells individually just grow in size. Their outer layer is called the hypodermis, and it's about 1 cell thick. It secretes a cuticle or an exoskeleton. And then another thing about the hypodermis is that it's largely syncytial, which means many nuclei share the same cellular space and cellular membrane. And underneath the hypodermis are 4 mononucleate muscle bands that run the length of the worm. They have a complete digestive tract from muscular pharynx to anus, a ventral nerve cord similar to Drosophila, which we talked about. As far as innervation goes, they also have tail ganglia and some innervation around the pharynx and also muscles surrounding the pharynx. They're pseudocoelomates, so their fluid-filled body cavity doesn't have a complete lining of mesoderm. Most notably, you'll find that the gonads are very large in C. elegans and take up most of the body mass. The gonads open up the midventral vulva, and this is a really popular topic in developmental research, which makes sense because they're very reproductively oriented. There are 2 arms to the gonad, and the germ cells are developing within the gonad's base. The germ cells that are furthest from the opening develop as oocytes, and the germ cells closest to the opening develop as sperm. And then the developing mature eggs that have been fertilized are in the uterus, which is closest to the gonad opening, or the vulva. The eggs themselves are fertilized on the way out, and then they mature to about the 30 cell stage before they're laid. One thing about C. elegans is that the shell on the eggs is fairly impermeable, so microsurgical techniques aren't common in studying C. elegans development. A lot of what we know about maternal effect genes comes from mutagenesis screens in C. elegans. Maternal effect genes control development in C. elegans up to about the 26 cell stage. Um, They discovered this shift from maternal effect to zygotic gene control by inhibiting RNA polymerase in developing embryos. Let's just take a second and think about why would inhibiting RNA polymerase be useful for determining whether or not gene control was happening zygotically or by maternal effect genes. So it turns out that RNA polymerase is necessary for translation of the zygotic genes, and that gets activated around the 30 cell stage. So if RNA polymerase is unable to transcribe genes and the maternal controls run out, then the developing embryo will start to have trouble completing developmental processes. Okay, so as I said, much of what we know about maternal effect genes comes from mutagenesis screens. Some examples of maternal effect genes that we found through C. elegans mutagenesis are polarization genes like PAR mutants and P granules, which we'll talk more about later in the lecture. But to start off, they use volvulus mutant worms that don't have a gonadal opening. And this is useful because viable offspring will actually just end up eating their way out of the worms. The eggs can't be laid since there's no gonadal opening, so it's a good way to easily screen for mutations that are lethal because those offspring just don't develop and then you don't really have to deal with them in the lab. Additionally, when using volvulus worms, it's easy to keep track of genetic lineages because there's no outcrossing involved. All of the reproduction is through self-fertilization, and you can easily maintain that because you know that there's no cross-fertilization if the hermaphroditic worms have no volval opening. One other thing to note about outcrossing is that it is possible between hermaphrodites, and occasionally there are males that arise within the population from non-disjunction of the sex chromosomes. So normally sex determination in C. elegans is determined by the presence of two X chromosomes, which leads to the development of hermaphrodites, but sometimes through a non-disjunction event, an XO male will arise, and they are sexually dimorphic in that the males are smaller and they have only a single arm to their gonad and they only produce sperm, and they can mate with hermaphrodites. Okay, back to the mutagenesis. So in the mutagenesis screens, the germ cells of a hermaphroditic parent generation are mutated, usually through chemical treatments or other ways of inducing mutations like radiation. Next, the mutated germ cells develop into adults, and then those are self-fertilized to give rise to the F1 offspring. And in the F1 offspring, typically there'll be one copy that's mutated, so you'll just have heterozygous for the mutation or homozygous normal hermaphroditic offspring. This is the generation where dominant mutations would show up, so you can screen for dominant phenotypes. But normally you will have to do another round of self-fertilization for recessive mutations to show up. And if they're maternal effect, then they won't show up until the F3 generation. So you have to do another round of screening. And this is exactly how they identified a lot of the useful mutations that they used to learn about C. elegans development and normal function. Okay, so now we're going to get into some of the specifics of development in C. elegans. Like we talked about before, larvae have 558 cells when they mature to adult hermaphrodites, and they have exactly 959 somatic cells. And researchers know the lineage of each cell. Every embryo shows the exact same sequence and orientation for every division. There's complete invariance normally in C. elegans. They hatch at about 14 hours, and as I said, they have about 558 cells at that first larval stage. The eggs are laid around the 30 cell stage, which is coincidentally when the zygotic genes start to drive development, which we talked about a little earlier. C. elegans do undergo gastrulation, but since we know the complete sequence of all of their divisions, we know the origin and fate of every cell, so it makes it a little less necessary to keep track of what the germ cell layers are doing, although there still is the process of gastrulation and a distinct ectoderm, endoderm, and mesoderm are set up. The first sign of anterior-posterior patterning occurs at fertilization. The sperm centrosome entry initiates the protein interactions that start to polarize the cell, and the cortical cytoplasm changes some of the concentrations that affect other early determinants. The site of sperm entry marks the future posterior, and that marks the anterior as well by default. After fertilization, meiosis completes, and then the shell forms. Meiosis in the oocytes is arrested prior to meiosis 1, and then after fertilization, meiosis 1 and meiosis 2 complete. And another phenomenon occurs called cytoplasmic rearrangement, where the internal cytoplasm rotates posteriorly and the cortical cytoplasm moves anteriorly, and both of these movements start to mix some of the determinants in the cell and set up polarization. The next stage is a pseudocleavage, where a furrow forms, but the cell does not completely divide. The first cleavage is initially asymmetrical, and that starts to also affect the future fates of those cells and setting up some different identities. The first cleavage gives rise to the AB cell and the P cell. And the AB cell and the P cell will continue to give rise to distinct daughter cells when compared to one another, and that will continue on to specify cell fates down the line. Okay, so C. elegans were some of the first organisms that we discovered cell polarization processes in. We analyzed loss of function polarization mutants, or PAR mutants, and discovered that PAR proteins and the genes responsible affect polarization in early cleavages. The sperm entry, as I mentioned before, causes a small change in concentrations, which further drives the mechanism of mutual repulsion between the PAR proteins 2 and 3, which we'll talk more about on the next slide. Another important determinant is the P granule. P granules are RNA-rich packets that are initially distributed evenly throughout the cell. They are maternal effect genes and products, just like the PAR proteins. And then after fertilization, they are moved towards the posterior of the cell through that cytoplasmic rearrangement, and they eventually congregate just in the end of the cell that will divide into the posterior most cell in the next division. Both types of maternal effect products, PAR proteins and P granules, are really important for cell polarization and setting up the fate of the next cells. So the presence of P granules will end up specifying the P cell lineage, and the absence will end up specifying the AB cell. The same thing goes with the PAR genes. So the cell that has mostly PAR3 will become the AB cell, and the cell that becomes the P cell will have mostly PAR2. The early regional pattern of the embryo arises through action of the PAR proteins, which become segregated along the anterior-posterior axis after fertilization, and these control the distribution of the other cytoplasmic determinants between the early blastomeres. Here is a graphic that depicts the localization of the PAR mutants throughout the first development and the moments leading up to that. Here the red shows the distribution of PAR3 and the green shows the distribution of PAR2. Prior to fertilization, PAR3 is distributed throughout the cell and PAR2 is not quite active. And this is shown in the cell in image A. Image B shows after fertilization that PAR3 starts to shrink its distribution back anteriorly, while PAR2 starts to become distributed around the posterior. The arrows in the middle show the directions of cytoplasmic rearrangement, and the blue circles are showing the nuclei that are getting ready to divide into the next cells. The central arrow is going to represent the internal cytoplasm that was initially part of the oocyte and its journey towards the posterior, and the outside arrows are showing that the cortical cytoplasm is migrating anteriorly and bringing with it some of the determinants that were present on the posterior end. Now, by the time that the cell completely divides, you'll see that the distribution of PAR3 is mainly in the cell that becomes anterior, and the distribution of PAR2 is mainly in the cell that becomes posterior. And this is important because they have different functions. In order to set up that gradient, they mutually repel each other to either end of the first cell. And then in Now, for our understanding, it is sufficient to understand that par 3 was functioning in mutual repulsion with par 2 and repelled it towards the posterior most cell. And we should know about par 2 is that it is actually a protein that functions in interacting with the mitotic spindle in order to shift it to a 90-degree rotation so that the future divisions in the P cell lineage are actually parallel to the first division as opposed to perpendicular in normal cell lineages. So the cell divisions in the AB cell and its descendants will be perpendicular divisions. And just to summarize again, the divisions in the P cells will be parallel divisions, and this is important in specifying cell fate. This is also where the PAR genes and proteins get their name because this is a partitioning function, and so when they are mutated, there were differences in division that were notable, and that's how they worked back to figure out what these proteins were functioning in and which genes were responsible. Okay, so as I said before, gastrulation is a little bit atypical in C. elegans, and the precision of the cell lineages makes it less useful to define which parts of the embryo belong to which germ layer. However, there still is a process of gastrulation that starts around the 26 cell stage, and at this point, 2 of the E cells move into the interior through an autonomous invagination, and this is followed by myoblasts and some pharyngeal cells. So the importance of this gastrulation still is in setting up the official germ layers, which are the ectoderm, mesoderm, and endoderm. And if you look at this chart here, this will show you a nice summary of which cells give rise to which other cells and which tissue types. And so if you look at the AB cell, for example, this is the ectodermal cell, and the origin of all the ectodermal cells, and it makes up it gives rise to hypodermal tissue, neuronal tissue, muscle tissue, and structural tissue. An example of a mesodermal cell would be the MS cell. The EMS cell divides to give you the E cell and the MS cell. The E cell produces endodermal tissues in the gut, and the MS cell gives rise to mesodermal tissue such as the muscle tissue, glands, and somatic gonad. Now, an interesting thing to note is there's a little bit of overlap between the tissue layers and the cell lineages. So, for example, the AB cell also makes a little bit of mesodermal pharyngeal muscles, and the MS cell also gives rise to some ectodermal pharyngeal neurons. However, for the most part, they are true to giving rise to individual tissue layers. And the importance is that we do know all of the details of the cell fates, so we know what to expect, which makes it easy to interpret when mutations and abnormalities arise and then figure out their genetic and protein origins. Another notable thing is that you can see exactly how many cell deaths are supposed to be occurring in each cell line, and these are developmental cell deaths, so not every cell that's generated during development actually will survive and be part of the adult organism. There are a total of 10,090 cells that are generated during development in C. elegans, but we know that there are only 959 somatic cells that are part of the adult organism, and so there are 131 regular cell deaths that are fated within this developmental scheme. Okay, so speaking of cell death, about 10% of the cells generated undergo apoptosis during normal development, mostly during a death wave, which occurs between 250 to 450 minutes of development, which is about 4 to 8 hours. Most of these cell deaths are regulated autonomously, which means the induction signals for cell death come from within the cells themselves, but some are regulated non-autonomously, which means the induction signals come from neighboring cells. So one way that they showed whether or not cell death signals for these cells that are fated to die come from internal signals or external signals was by ablating neighboring cells or removing them from cells that they knew were fated to die, and then determining whether or not that impacted their cell death fates. So if removing neighboring cells caused cells to live that were normally fated to die, then that would mean the cell signals were non-autonomous and that those neighboring cells were required for cell death to occur. And if there was no impact on cell death fates by removing neighboring cells, then that would indicate that the cells were autonomous and internal, and that those neighboring cells weren't required for cell death to take place. There are some genes that we know are part of the conserved cell death pathway, and the way we figured this out was by observing mutants for cell death. So since we knew which cells were fated to die, we could tell how they genetically were different, or protein expression was different between the cells that were wild type and the cells that didn't die properly. So the cell death defective mutants, or SED mutants, are all part of the conserved pathway, and they affect all cell death within the cell. And while there are other mutations that affect cell death only in specific cells and cell types. So the cell death defective mutants, or SED mutants, were discovered in cells that we understood were supposed to undergo cell death normally throughout division, and then they had abnormal phenotypes for cell death, and we worked back to figure out that there are these SED mutants like SED9, SED3, and SED4. And these genes all act in the same pathway. In SED9's normal function, it inhibits cell death by inhibiting the activity of SED4, while SED4 activates SED3, which continues the cell death pathway and activates the cell death processes. So, removing function of any one of these genes would result in abnormal apoptosis or cell death. And from the graphic, you can see that a SED9 mutant or loss of function would result in more apoptosis than normal, as it's normally functioning to inhibit cell death. A SED9 gain of function would result in a downregulation of apoptosis, or fewer instances of cell death, because it would be overly inhibiting cell death. And then either SED3 or SED4 loss of function would result in a lack of apoptosis or no cell death, because they are meant to activate the pathway, and they must both be present for the pathway to activate. Okay, so let's take a closer look at what the actual functions of the SED genes are, and how those compare to mammalian cell death pathways, because as I said, C. elegans is a model organism that helps us figure out a lot about other organisms, because they have very similar functions and homologous functions, but they are simpler to work with. Okay, so SED9 is a homolog to the mammalian protein BCL2, which is a mitochondrial interaction protein that inhibits cell death. So the way they have it represented here, BCL2 is on the membrane of the mitochondria, and it actually binds to SED4 and keeps it inactivated. SED4 is homologous to the mammalian APAF1, which is part of the apoptosome that promotes cell death in mammals, and it does the same function in C. elegans. While SED3 is homologous to caspase 3, which they are both caspase proteins that activate other caspases, and they also promote cell death and enzymatic breakdown of the cell membrane and other cell parts. So when a stress signal comes in that there's DNA damage or some other factor in influencing cell death, then EGL1 is activated, and what EGL1 does is it actually binds to SED9, and SED9 then releases SED4, and when those SED4 proteins are free, they actually assemble into the C. elegans apoptosome, or the spinning wheel of death, which then activates SED3, a caspase, and promotes cell death. This is very similar to mammalian cell death, where the BCL2 actually activates cytochrome C. There's some intermediate steps, but the cytochrome C ultimately activates the apoptosome, which leads to caspase activation and the degradation of nuclear membrane proteins and targeting cell repair mechanisms. Okay, so this next figure ties into the journal article that we're going to read for our next lesson, and the important parts that I wanted to highlight here is that we're looking at cell death, but it's in a different pathway. So before we were just talking about general cell death patterns in development, but this article looks specifically at cell death in germ cells that's activated by DNA damage. So I just want to take a moment and think about why we would need cell death pathways that are activated by DNA damage and what type of impacts that would have moving down the line if we didn't have DNA damage-induced cell death. Also, I wanted to highlight that the conserved parts of the pathway should look familiar to you because we're discussing them in this lecture, and that should be helpful in orienting your readings moving forward. Now, to go back to the importance of having a DNA damage-induced cell death pathway, DNA damage in the germline is particularly problematic because it can be passed on to the next generation. And so that would form whole organisms that are starting with damaged DNA or possibly mutations that could be harmful, or mutant phenotypes that could cause issues down the line. And having cell death pathways that are DNA damage induced just in general in all cells is important because it is a defense mechanism against conditions like cancers and other degenerative disorders that can affect the entire organism. So this article in particular looks at the function of one gene called KRI1, or CRE1 is how I'll pronounce it. So in previous screens, they came upon a mutant phenotype that resulted in a decrease in cell death that should have been initiated when DNA damage was induced, and that is how they figured out CRE1 functions in the cell death pathway. And to further investigate this, they did some studies on how CRE1 fits into the cell death pathways and trying to figure out what its actual function is and whether or not it's regulating cell death in an autonomous way or in a non-autonomous way. And in this study in particular, they invoked ionizing radiation to induce mutations in DNA in germ cells and also in some somatic cells for comparative studies. And I just wanted to give you a little bit of background on ionizing radiation before you read the paper. So ionizing radiation is just a technique for mutagenesis where gamma rays or X-rays are used to create double-stranded breaks, single-stranded breaks, and even base pair changes in DNA randomly. And for the author's purposes, they are inducing radiation to try to activate the DNA damage-induced cell death pathways, and then they do some further comparisons to figure out what's going on with CRE1 function and how that relates to other genes that are known to function in the pathway. The last thing that I want to talk about is another technique for the paper that is a mechanism for RNAi interference, and I just thought this was a neat technique that might not make sense right off the bat, so I want to describe it a little bit further, but it's how they managed to knock down CRE1 through RNAi interference, which in this instance, what they did was they engineered plasmids that had double-stranded RNA endogenous to the CRE1 sequence, and then they put those plasmids into E. coli through bacterial transformation, which is something that we've done in lab in a different way for a different purpose. And then they cloned those bacteria and actually just fed them to C. elegans as their food source. And then at sufficient doses, the endogenous RNA will actually be knocked out by that double-stranded complement. And it's even strong enough to knock out gene function in the F1 progeny of those original worms. So it takes about 36 to 40 hours for larval worms, and then you transfer some of those adult worms, and it takes about 24 hours until eggs are laid, and then they remove the adult worms, and then in about 24 to 72 hours, they should be able to see the knockout phenotype in the sample worms.